Let's go ahead and get started. I would like to go back to some of the slides that we were working with last time. And so if you've forgotten how to find those slides, there's two different ways that we can use. Come with me, if you will, to the website. And you can go down to the bottom. Oops, sorry, not here. You have to go to the class. You go to Bioinformatics. And then you scroll down to the very, very bottom, and you should find your slides here. Slide 2. I'm thinking that this may be the last time that I put up slides on the website, because I'm hoping that everyone here has installed Git by now. And if you have to make an account on GitHub, then you please do that. You may have to do that. And I think you do have to make an account. For these, actually, to pull over these slides, I don't know that you do need an account, but when you're pushing your work for your lab, for instance, you will need an account on GitHub. And for doing, when you do that, please use your Allegheny email address, and that will allow you to use the classrooms that I've set up for this class. Otherwise, I don't think it works. There's something strange about that. Anyway, so let me go ahead and just check my notes, make sure I've got everything up here that I want to show, and we'll get started. So let's go back to the slides, and we'll go back to where we left off. So hopefully you can. Oh wait, sorry guys, you still can't see me in the room here. Let me just move over. Okay, hopefully you can all see me there. Yes. Okay. So these are the slides we were working on the other day, and the slides we ended up where were we here? We spent some time talking about careers. I do hope that you spend some time actually thinking about careers in bioinformatics, and that is because there's a lot of stuff out there. There's a lot of things that you can actually do to start up your career, and I think that you might actually enjoy the challenge. I think that when I first heard of bioinformatics, I thought, wow, what an amazing area. There's so much science that isn't yet known, or so many things that aren't yet known in bioinformatics, so many problems yet to be worked out. It just sounded absolutely delightful. And so, and it was, and it is. And so I would recommend spending some time looking at this. If you're looking for a career area, then by all means, check out bioinformatics. There are many, as I call them, low-hanging fruits, like projects that need to be done that just haven't yet been worked on by anybody. Anyway, so we began working we began looking at like the background of biology. That's one of the things that we have to do in this course. A lot of people who've taken this course before, they haven't necessarily been biology students, and so I spend this time to talk about like what the biology is behind the work that we do. We do get into the computer science part, and in fact that's going to happen very, very soon. But for now, though, I wanted to spend some time just kind of giving you an idea about what is this thing that we call DNA? What is deoxyribonucleic acid? I mean, what does this stuff do? What is this genetic material supposed to be? What's it supposed to do? Look like? How does it work? And so I wanted to spend some time just talking about that. Now, this was probably one of the slide that you see now on your screen is probably the most important slide, or the most of this whole PDF document here. But it's probably the most important concept in biology, hands down, the most important concept in biology. And that is because every single thing that happens in biology is really building on this premise, and that is that DNA gives us RNA, which gives us protein, and then protein is then used to build things that, like your skin and your hair and bone, and seemingly everything that you need to use comes out of protein. And so this is actually a very important slide. And so every single thing that you'll see happening in biology and in bioinformatics supports this dogma. We're getting some blind feet on our overhead projector here, but everything that we do supports this dogma, and that is that DNA gives us RNA, which gives us protein. And so that is probably the big piece. So we discussed that DNA is this stuff that lives inside the nucleus of a cell. It's probably the most important. It's the most coveted thing that a cell owns. But to the cell, though, this is the most valuable thing that it owns. I mean, it's the most valuable thing. It contains the genetic code for that cell, and without this thing, the cell can't do anything. It's like the brain for the body, for instance. Without that brain, the body can't function, and without this DNA, the cell can't really function. And then we discussed kind of like how the structure was discovered by Watson and Crick using the research from Rosalind Franklin. where she was working with X-ray X-ray crystallography. But the most important thing, though, is that the structure itself is a real marvel. I mean, the way that DNA comes together. In the middle here, you have we discussed these rungs to this ladder. Those rungs are the I guess you can call them those are the bases here. Those are like where you put your foot if this were a ladder. But these are the bases. And notice that A's and T's attract each other. So A is the complement of T. And G's and T and C's attract each other. They are the complement of each other. So A is adenine, thymine, and T is thymine. Guanine for G, and C is cytosine. And so these are these bases, or the base pairs as we call them here, that attract each other. So T is the complement of A. That's how you'd say this. That means that the T connects to A. But one thing, though, that I want you to look at is in this diagram. I like how they've drawn this diagram. You'll notice that how the T's and A's connect to each other is a kind of a jigsaw puzzle piece connection. And it's a different, and so is G and C. But you'll see that G's and A's cannot connect by the geometry of these connections. It will never happen that way. You'll never get a G and an A connecting. And so this is actually something that happens in nature all the time. So in order to prevent G's and A's from connecting, or T's and C's from connecting, I mean, I say to prevent, they don't connect because of the actual chemical makeup that exists between G's and A's. It's like they don't conform. They don't connect chemically. And so if you look at this design here, when you, I'm saying this because when people see these types of designs, they imagine that A's and T's kind of slot together like jigsaw puzzle pieces. But that's just designed here to show you that A's and T's are complementary bases of each other. But when we look at the purines and the pyrimidines, somewhere, which we'll see later on, we'll actually see that the actual the molecular biology or the organic chemistry side of DNA, of the base structures, the molecular model of the bases themselves, actually kind of you can't connect two pyrimines together or two purines together, and we'll see that in a moment. So we'll get to that in just a second, but we're really talking about the structure of DNA, and your DNA is really composed of your bases, and then you have these phosphates, which are there on the sides. You have the deoxyribose sugars, and the phosphates that are on the kind of the outside of the ladder. And on the rungs, we have the bases. And then when the DNA is actually kind of in its stable conformation, or its stable state here, it's kind of twisted on itself. And you can see that this right here, this picture is where the DNA is kind of like twisted. Now, this is interesting about the twisting because you'll notice that you have these, in this diagram here, you have the major groove, and then you have the minor groove. You see that in the middle of the DNA? You'll see that there's actually more space between them in the major groove, between one edge, I'll call it the edge, but one of the sides and the other side. It's like the distance between those objects, those molecules on the edge, is greater in the major groove, and the minor groove, they're less. And you can kind of get an idea about how that groove happens. That's just a ladder, wherever it was. That's just a ladder being twisted here like this. But because you have this distance and space in the major groove and minor groove, that's actually central. That's actually very, very important. Because if you didn't have those grooves in that particular space, and this is something that's really, I find just super exciting, you wouldn't be able to have certain types of proteins latch onto the DNA to do their work. So for instance, that's how these proteins can feel themselves around or feel their way around in working with DNA. When they read DNA, or when they copy DNA, or when they're unraveling DNA, when they're repairing DNA, when they're doing these things, something has to stick to this twisted helix, unravel it, and then do some work, and then ravel it back again. And when something first latches onto the DNA, it knows where to latch on by whether it's the big groove or the little groove. Anyway, that's actually quite exciting. There's a couple of videos at the very, very end of these slides where you can. Where they talk about that in more detail, and they have some computer graphics to show you how all this stuff comes together, and it's just incredible. I mean, it's just incredible when you see how this, you see how the DNA works with all these different agencies that come together that manage it. Wow, it's just exciting stuff. Very, very exciting. Anyway, so moving on. So then we were talking about the nucleotides. Now, the nucleotides are this area, these things that hold the bases. So you can think of them as the ribose sugar and the phosphate groups, and the bases. This is all this. It's the supporting rungs of the ladder, or the base pairs. You can think of all this kind of stuff as coming together as kind of like the meat of DNA. But the nucleotides are composed of these three subunits: the nucleobase, which is this thing down here, which is like the A, the T, the G, or the C. Then we have this 5-carbon sugar, which is this purple flying saucer thing. I call it the flying saucer because it kind of, when I first saw these molecules, I thought it looks like some kind of spaceship or something. It's actually a ring from organic chemistry. And then we have a phosphate group consisting of 3 phosphates. Actually, not 3 phosphates. Sorry, it's 3. It should be. It's 3 oxygens here, but it's 1 to 3 phosphates, and this one you have 1. But you have these. You have this kind of this phosphate group. You have this sugar, and then you have this base, and that's really the essence of DNA. The interesting thing about this is that when they found all these things in DNA, when early biochemists took some DNA and then they ground it up. That's really one of the big experiments that they do in biochemistry in the wet lab. If you take some protein and you kind of like put it into a micro blender, just grind it up, just grind, and you see what kinds of things float to the surface. So, for instance, if you're making like a strawberry smoothie, for instance, you put your strawberries. Imagine you didn't see all the stuff that went into the blender, and you just push start, and then suddenly you start smelling strawberries. You'll think, oh, this must be a strawberry smoothie. There must have been strawberries in here because I smell strawberries. Well, the same thing happens with DNA when they were working with DNA in the beginning, and other types of proteins too. They would just grind it up, and they would see what kinds of things like float to the surface, and see that, oh wait, there's this in there. Well, I guess that must have been a part of DNA. I didn't see that before, but I see that now because it's there, and there was nothing else there before, so it must have been from the DNA. And so they were able to isolate some of these things. But the real issue here was, how does all this stuff come together? And that's why we're talking about Watson and Crick and Rosalind Franklin, because they were able to figure out how all this stuff connects. In other words, they had all the pieces of Lego, but they didn't know how the pieces came together to build the helix. And that's what their work was all about. And so this is really kind of getting into their work. The DNA structure, for instance. And how do we know that DNA looks like it and how it does? Well, because they put together a model, they tested the model, and this is the only model that survived all of the tests. And every time they test it today with different types of, every time they test DNA, the structure of DNA with new tests that are coming out today, it only works to confirm that the structure is the same one that they discovered back when. This is actually important. One thing that I was talking about with this DNA structure is, notice how I'm looking at the gray diagram on the right-hand side, and you'll notice that in the sugar phosphate backbone on the left-hand side and the sugar phosphate background on the right-hand side, one side appears to be upside down. Does everyone see that? On the right-hand side, it's like the S's or the sugars appear to be upside down. That's not a typo. That's because you have DNA which has a specific direction. There's what's called a directionality in DNA. And you'll notice that on the left-hand side of this gray illustration, the direction is that you have a phosphate that connects to a sugar, then a phosphate, then sugar, and blah, blah, blah. And the sugar has this point that points upwards, whereas on the other side, the DNA, the other strand of the DNA here, that sugar is pointing down. This is what they call the 5' and 3' direction of the DNA. And to explain how that works is, you'll see that in this, I'm looking at the deoxyribose molecule in the middle. You'll see that there's like a 5, little mark next to it. That's 5', 4', 3', 2', 1'. Each of those primes is next to a C, or a carbon. And so in molecular biology, you have to give a kind of an address, if you will, to each of these molecules, so that you know which carbon is it that actually is affected by some reaction. Now, in this case, DNA has this 5' carbon, which is affected by a reaction, and a 3' carbon, which is affected. Now you'll see that in this deoxyribose, I don't know if I can click on this stuff here. I can't really click on it, but I wish I could. But on the 5' carbon and the 3' carbon, if you look at where those carbons actually are in the ring, you'll see that that same carbon in the diagram in the gray has a phosphate group on it, on the 5' carbon. There's a phosphate group stuck to that carbon. Does everyone see what I'm talking about? Look at the S. There's that ring, and then you'll see there's a line that comes up. I'm looking at the left-hand side of my illustration here, but on my ring here, my sugar ring, there's a little line that comes straight up. That's a bond, and then it bends. There's like an elbow, and then it makes its way to a phosphate. That bend is a carbon. That's how you draw out in organic chemistry that carbons exist, because there's a bend in that line. The line is the bond, and the bend is the carbon. If it wasn't a carbon, they'd actually write in what that atom was. As you can see, they've put in like oxygen, hydrogen, and other things. But typically, though, you wouldn't put C, because it's organic chemistry, and organic means it's carbon. So anyway, what I'm saying all this, though, to say that there's a directionality in DNA, and that is that when you're building DNA in this, you can see in this diagram here, you're building onto the 3' carbon. That means that in your S ring, you can see that lower left bottom corner of the sugar has a line that comes out of it, a bond that goes to another phosphate group, and every single sugar you see there has a similar line at the same location going on to a phosphate group. So when you're building DNA, or when you're thinking about the directionality of DNA, DNA is actually built in the direction of 5' to 3'. That means that you first start with, you'll connect some phosphate group where you start. That would go to your 5' carbon. That would connect to the 5' carbon of your deoxyribose, and then to build the next phosphate, like as you're moving downwards here on this left-hand side, then you build that next phosphate onto the 3'. So really what this is saying is that if you are a sugar group in DNA, you have phosphates on your 5' carbons and your 3' carbons. And that is absolutely essential here, because as we see in a moment, this will describe how you can replicate DNA. If you didn't have that, you wouldn't be able to replicate. This is kind of, if you think of it in terms of kind of I don't know, maybe a GPS in molecular biology, this is helping to form a direction so that the molecules themselves know how to build themselves, how to stack one complex onto the other, or one molecule onto the next, onto the next, onto the next, to build out that DNA. And you would be building out that DNA if you are working with, if you're copying a strand of DNA. That's really how copying works, and that is absolutely an amazing process. We'll get to that in a second here. One thing I'd like to say, though, is that remember that AT, the A's and T's attract each other. There are 2 bonds in A's and T's, only 2. And there are 3 bonds in G's and C's. There are 3 of them. And so G's and C's are stronger bonds. They're bonded together more strongly. It's like having 3 magnets holding things together rather than 2 magnets. And so G's and C's actually do strengthen, if you will, strengthen the DNA. And in certain studies in DNA, you will find that in parts of DNA which are prone to being under a lot of, they call like agitation, where, for instance, maybe a piece of DNA at particular regions of the code where they might get bumped and frazzled and pushed around, they tend to be built out of GC bonds because that's tougher, holding everything together. So it's like, imagine like it's like a little person building DNA, and they're saying, oh, well, this part of DNA is probably going to get bumped around, so I better make it strong, so I'll put a lot of GCs in here. And it just kind of happens that, it just comes together this way. And it's just, I mean, it's all completely random from nature and driven by environmental pressures. But the way it comes together, though, is just incredible. I mean, it's just incredible. I think that this is really the last slide about where we stopped. I don't mean to keep taking everyone's time on these on the background here, but this is actually quite important. We're talking about purines and pyrimidines here, pyrimidines. I keep getting that word wrong. But these are different types of bases that you have in DNA, and so a purine is going to be attracting a pyrimidine, and that's kind of how it works. It's like the pyrimidine is the complement to the purine, and so it's like they connect each other. But what I wanted to say, though, is that if you look at, I'm looking at the box here that has the orange box. You'll see that that's A and T, And you can see that the two bond, or the double bond for AT can be explained by how the molecules look. And there's no other way to connect these things together such that you get 3 bonds. And so that's how you know that A's and T's only have 2 bonds. But now look at the green bar, or the green box just below it, where you have a purine and a pyrimidine coming together. That's G and C. And it has you have 3 bonds. You have 3 bonds that connect those 2 molecules together. And so that's how you know that, for instance, by looking at the organic chemistry, by looking at the model of the molecular model of these bases and their connections, that's how you know how many bonds there are. Now, I'm saying all this because when you are working in bioinformatics work, when you're putting together some kind of a model where you have a computer where it's running some kind of an algorithm that's doing something that's happening in nature, you have to be well aware of these kinds of details, because your model has to reproduce this kind of work in silico. That means you're creating a piece of code that's going to be kind of working with DNA, or it's modeling something to do with DNA, and it has to be following this model. You can't do any other model, because otherwise it might be a cool model to watch, but it's not going to be realistic in terms of like what's going on in nature. And so this is just up here to show you that you really do have to know some of the detail. But in this course, though, I'm not going to be harping on about all this background in biology forever. In fact, we're going to be going on and doing some coding very, very soon. I think next week even, we'll get into the actual computer science part. But I think that this is still important. This is another slide where we come back to this prime and 3' directionality of DNA. Again, you can see another picture where this purple spaceship ring, that's our sugar, is connected on top to a phosphate group on its 5' carbon, and then after that, the next phosphate group is connected to that particular sugar's 3' carbon. And that's how things build. And so the directionality here is 5' to 3'. In other words, you're starting at the top of this molecule and working your way down. And so if you are reading genes out of this DNA, you would start by saying, okay, well, this is my first base is A, then I'd see G, then I'd see C, and then I'd see T, and I just keep on going down, 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 down. This is on one of the rungs. Now, on the opposite rung, you would be going up, 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 because again, you can only move in like the 5' to 3' direction, and so therefore you'd be looking at, you don't see this complementary strand there. But if I were starting at the T on this strand, the complement of T would be A, and so I'd be starting at A and moving up to the next one, which would be, well, what would the next one be? If we have C over here, what would be the complement? Yes. So it'd be G. It'd be G. And so if we're going if we're on the other side of the sequence, we're at T, then we move to G, and then what? What do you guys see? What's the complement of? I know you know. What's the complement of G?
C, right?
It's C. That's correct. That's correct. Thank you. So then what I'm saying is, like, you're reading DNA in this direction like this on one side, and then on the other side, you're reading the DNA going up. And so that is the complementary. And so that, what I'm saying is that the directionality of DNA is always 5 to 3'. So you're reading or copying or transcribing or whatever it may be, but you're going from 5' to 3'. It's like, for instance, when we read texts in our language, in English, we start at the left-hand side and our eye moves to the right, and that's how we make sense of our words. If the words were backwards, it wouldn't make sense to us. And if DNA were backwards, it wouldn't make sense to the molecular machinery that's taking care of these. So this is actually an important discovery in molecular biology, in that you have this directionality. If you don't have this directionality, then you can't, your DNA could not be copied. This is just another picture of this 5' and 3' ends. One thing I'll move on, though, we'll talk about copying in a moment here. But before we do, there were 3 proposed mechanisms of how is DNA actually copied. In computer science, you can copy a file, just type CP space file name space new file name, press enter in your Unix machine, and boom, you've got 2 files. And one of them is a direct copy of the other. In DNA, though, you don't have a command that you can just type in like that. In fact, you have machinery that does that for you. And the machinery is remarkably efficient at doing this. I mean, it has spell checking in there. It has special types of stability checking and make sure that everything is actually connected the way it's supposed to be. It has like other types of fail-safe types of systems to prevent faults from happening during DNA. In fact, did you know that if your DNA that is trying to copy has a fault, a misspelling, there are certain types of agencies that come through into into this copying process that will say, "Oh, that's a spell check. That's an error right there. I got to fix that," and it fixes it, and then you don't get cancer. It's just incredible. It's incredible how it works. I mean, you have all these pieces coming together to prevent spell checks. Because if you have a spell check problem in your DNA as you're copying it, it's like your proteins don't work anymore. It's like, what would happen if you're working with code and you just like start pushing some buttons and I don't know what's going on here. Is it going to compile? No, it's not going to compile. You're going to have runtime errors. You're going to have all kinds of, or you may have logical errors. In biology speak, that means you have an ailment or some kind of disorder or cancer or something that's gone horribly wrong, where it's your system is making bad proteins. Anyway, so a lot of people were worrying, like back in the 60s and soon after the development of DNA, I guess 50s and 60s, how does DNA replicate? And so they had three main types of processes which they devised, and they found out that one of them actually works. You can see the happy face is the one that actually works, but I wanted just to spend some time to talk about what these models are. So the first one is called the conservative model, and the conservative model is a model where the DNA, in order to copy it, both strands come apart. That's how it works. Both strands, the DNA untwists, and each thing comes apart like this. And then each strand becomes a template. It's floating around in the ooze of the cell or wherever it is. And then it becomes a template for other little bases to start being brought in by molecular machinery to build the other side of the DNA. That's how it works. And so you have a template, and then you use that template to create another side of the complementary side of your DNA. And then after a while, you have another piece of DNA, which is exactly a copy of it should be an exact copy of the original one. I mean, sometimes it isn't. But anyway, then you have your DNA comes back together again, and then you have this copy. And then again, when that DNA is going to copy itself, piece kind of separates. They never come back together again. And then you have another on the left-hand side here. You have a complementary sequence that comes over here, and on the right-hand sequence piece, you have another complementary sequence that comes. And then you have then you have more DNA. Then you have another helix of DNA. And so that's really what we see in, for instance, in the semi-conservative model. In the conservative model, this is saying that the same two original pieces of DNA somehow find each other in all this mess, and they stick back together again. That does not happen. When the DNA comes apart, its pieces say goodbye, and those particular pieces never actually come back together again, because they are used to create complementary sequences, and so they're paired already with other pieces. And so it's impossible for them to come back together again. So it's kind of like you say goodbye to your complementary sequence, and you say, okay, it's been great to be with you. Take care, and then you separate, and then both you go off and make your own family, and the other one goes off and makes its own family, and you'll never actually become a family again, if you think of it in terms of family. And so the conservative model doesn't work, because there's no information to suggest that the two pieces can ever come back together again. So you have your one sequence of DNA, it untwists, you have your one helix, it untwists, two different sequences floating around in the ooze, they start attracting, or the molecular biology starts putting, or the mechanical, or the molecular mechanics start putting together all the bases and building the other side of each of the strands of DNA, and then they twist again, and then you have two new helixes. The semi-conservative model is what I'm talking about, where the DNA comes apart, each piece is out, and then each piece attracts a complementary side, a complementary strand. That's all done by the molecular machinery that's out there. I mean, there's a bunch of work that happens in that, but in essence, this is what's going on. So there's a semi-conservative model. Now, the dispersive model is a little bit different in that the dispersive model is saying that you have this, you have a piece of DNA which somehow is broken up to little pieces, and then those little pieces somehow attract other pieces, and then somehow those pieces all know where to go back into the sequence of DNA. Now, while that was actually a very attractive idea for some time, it really was not feasible, though, because there was no information, or there was no reason to believe that each piece of DNA floating around had any idea where it was supposed to go. Like there was no markers of like, I'm supposed to be in the first part, or I'm supposed to be in the second part, or I'm supposed to be in the third part of the DNA. There's no information about the order. Hang on a second. So all that stuff was there, and so you have this. You had this a lot of research groups going into looking for this thing, but they never actually found it. So anyway, what I'm trying to say though is that you have a lot of there's a lot of information, or a lot of interest in trying to find out how DNA is actually copied here. But in the semi-conservative method of DNA copying, you have this it's a rather interesting thing here. But you have this system here where you are working with a piece of DNA like this. And so what happens, maybe this isn't the best picture. This might be a better picture to start off with. But basically what happens is when you're copying DNA, think in terms of the directionality that we were just talking about a moment ago, where you have 5' and 3', where, for instance, you can only read, or your DNA can only be built, I guess, the directionality can only be read in 5' to 3'. And so what I'm looking at now is the upper strand in this diagram, where I'm looking at the darker blue color, and then there's like this lighter blue strand called the leading strand, which is being built onto the bottom of that. So really your DNA is like a zipper that undoes, and then on the side over here that's on my left, your right, and the other strand of DNA, then you start building in the new pieces of DNA, or the new fragments of DNA, to complement each of those strands. And after a while, this DNA kind of just comes apart, and then you have two helices that come out of this. Now, I'm kind of glossing over a bunch of things that are happening, but in essence, this is what's going on. But you'll notice, though, that getting back to the directionality conversation that we had earlier, you have this. You're building your DNA in the 5' to 3' direction. Well, how do I know that? Well, again, I wish I could point to some of this stuff, but maybe I can't. The other strand that we see here. Now you see that word where it says DNA pol 3? So right around there, that's a mechanism, or that's a piece of biomechanical, I guess you can call it, or maybe biomolecular machinery that is finding arranging the bases onto the DNA as they should, the complementary bases onto the DNA once it's been kind of unpaired with another piece of DNA. So the DNA unzips, you have each of these strands floating around, and then you have this molecule that sticks to that, each of those strands, and then it starts putting in place the bases, which are complementary to the bases which are already on that strand. This is what they call during the replication fork. And you'll notice that when you're putting in these pieces of DNA, you put them in a particular direction, and that's going back to this 5' to 3' directionality conversation that we had earlier, that we're building from the 5' carbon, which is that carbon above, that space where the carbon is, where we're going down. We're building going downwards. In other words, we're building, if I can just show you again, here we're building when we start on the left-hand side of this gray, from the top going down. That's in your 5' to 3' direction. And when I say building, what we're doing is we're actually just adding in the complementary bases as we go through. And so this DNA pol 3 is actually going through and figuring out what should be there. Here's my base. It's a T. I need to find a complementary or an A that goes right here, and it does that. And in doing so, it builds the other side of the DNA, or the other strand of the DNA. That's easy. That's on the 5' side of or strand of the original DNA. But on the 3' strand of DNA, this is where things get interesting, in that you can only build from 5' to 3'. That's the problem. And so what happens if you have if you're building if you're on and looking at the lower DNA strand, where we have these little red fragments that are being put together, or the Olazaki fragments. Those we have to, when you break apart DNA and you're building, and you don't have the luxury of building in 5' to 3', but rather you have to build in the 3' to 5'. Well, guess what? You still have to build in the 5' to 3' direction, but rather you build in little sections, little chunks in the DNA, and that's what we see at the bottom of this DNA that's unraveled, where we have the DNA pole 3, again, on the bottom strand. You can see those chunks that are being put in place, Okazaki strands. So you can see them right here, this Okazaki strands. This is in the middle of this graphics here. You can see that these, what is this? This is on the 3', the strand that's labeled 3' here. You can see that you're just building in little tiny pieces and putting them in place. Well, the reason why you have to put in pieces here is because each of those pieces was actually originally created in the 5' to 3' direction. You have to go 5' to 3' direction. And so if you don't have the luxury of going 5' to 3' direction, kind of like following the zipper all the way through, then you have to like say, okay, well, I'll just take this chunk, and I'm going to start at the 5' and work my way to 3'. Then once I finish this piece, I'm going to superimpose it into the sequence that I'm working on, and I'll just repeat and repeat and repeat. And so that was like a major conundrum. Or researchers were looking at these little pieces, including this professor called Okazaki, who these were named after, the Okazaki fragments. And they're wondering, why does DNA replicate in this way? How extraordinary. Why would they do that? Well, that's because you can only build in 5' to 3'.